Okay, for the enzyme lab makeup for the biology class, regular biology, I'd like you to start by reading the introduction. And then once you've read the introduction, we are using the enzyme catalase. Catalase can be found in all body tissues, and but especially we can find it in things that have a lot of blood, and numb blood has catalase in it as well. And so I just went over to Dillon's and bought beef liver, and we can find a source of catalase using beef liver. It's a little gross, but it's still a good source. Okay, and let me get a paper towel. So once you've read the top introduction, you can see that we make a chemical in our cells called hydrogen peroxide. You can buy hydrogen peroxide over at Dillon's in this brown bottle. Hydrogen peroxide is constantly breaking down, and even after I put it into this little cup, it still starts to break down, but breaks down really slowly. We make hydrogen peroxide in our cells, and it's pretty strong, and if it wasn't broken down in our cells, eventually it would destroy our body cells. And so we need to be able to metabolize or break that down into safe products. And you can see by the reaction on your lab that it breaks it down into H2O, which is water, and O2, which is oxygen. And so we can find this source of catalase that will help to speed up the reaction of breaking down hydrogen peroxide into water and oxygen. And so to start out with, we are going to look at the scale that's about halfway on the down the page. Looking at, if there's no bubbles, we would put a 0. But if there's bubbles that come up and over the test tube, then we're going to call that a 6. So everything else is kind of in between. So it says, for the first part, that we're going to combine hydrogen peroxide and liver. So I'm going to put some liver into the test tube. So I'll put a piece of liver in here and then push it down with a straw. So it's at the bottom of the test tube. And then I'm going to add a pipette full of hydrogen peroxide. And put it back in here. And we can see that there is definitely a speeded up reaction. Hydrogen peroxide is breaking down rather rapidly into water and oxygen. And I would say, I'll let you decide what you want to put down for the reaction. Look at the scale and how you want to determine that. And then it asks you what happens if you add more. So you put the reaction number down for number 1 and then describe what's happening. And once you've done that, then we can look at number 2 and ask ourselves what happens if we add more hydrogen peroxide. Does the enzyme continue to work? So after looking at this, it asks you on number 3, what can you say about enzymes? Do they work one time and then stop? Do they seem to never stop working? Or something in between? So can you describe how you would see enzyme activity? And then you can turn your page over and it asks you, what evidence do you have? So whatever you said in number 3, what evidence do you have to back that up? And if you don't have any evidence, maybe you need to look and say something different about enzymes. Think about what we've seen, what we've learned so far about enzymes, and then what evidence do you have that that's happening? All right, now we're going to look at what happens if we change something about the enzyme's environment. Today we're going to change the temperature. So instead of it being room temperature like that one was, we're going to use a small piece of cooked liver. So I boiled some liver here and put that into a test tube. And once again, we'll use a pipette full of hydrogen peroxide and look for some reaction. Okay, and you can decide what the reaction number would be for that one and describing its activity. High temperatures tend to change the shape of the enzyme so that it no longer fits the substrate that it's trying to break down. That's why doctors become very concerned when people have very high temperatures. But that's also the premise of why we cook, so that we can change the shape of those proteins and make them more edible for us to eat. All right, let's try a piece of cold liver. Down here. There we go. I just wanted a little piece, and of course a huge chunk was coming. Okay, and I'm going to go ahead and stick that down with a straw. And I'll add another pipette full of hydrogen peroxide. Ooh, ooh. Where did that go? So can you describe what's happened here? Get a little idea of what's left behind back here. So comparing these two right here, being boiled and frozen, you can see there's a pretty big difference. And I want to know then on number 6, what temperature works best for catalase? At the very end, you're going to finish up by telling me what you've learned about this enzyme catalase, this enzyme that we find in liver and other tissues of our body. We all have catalase in all of our tissues, in all of our cells. But who doesn't? Bacteria doesn't. So when you use this to clean out a cut, you probably have noticed that it bubbles, and it is killing any bacteria that isn't able to break it down because it is toxic to the bacteria. So it makes a pretty good antiseptic for humans to use. All right, and I'll let you finish filling out your lab, and you may take pictures of both sides and submit that to classroom.